Hi class, Dr. Linder. Okay, in a previous video, we've talked about neurophysiology. Now let's talk about the propagation of the action potentials. So, um, an action potential spreads or propagates over the surface of the axon membrane. So, as sodium flows into the cell during depolarization, the voltage of adjacent areas is affected and their voltage gated sodium channels open, creating the self-propagating effect on the cell membrane. And that traveling action potential is a nerve impulse. What can affect these nerve impulses? Well, we can do things like try and prevent the opening of the voltage gated sodium channels. We can prevent them or slow down the opening because it's the opening of them that creates the action potential. So nerve impulses cannot pass an anesthetized region. And that's why Novocaine or lidocaine is used during dental work. We don't want to feel the pain. We don't want those nerve impulses or action potentials getting up into the brain. So we use these local anesthetics. You can also use ice. Ice doesn't prevent the sodium channels from opening, it just slows them down. Okay, so you could use ice, you can use Novocaine and lidocaine. Now, there's two different types of conduction. There's continuous conduction and saltatory conduction. And of the two, the faster one is the saltatory conduction. Continuous is a little bit slower. Continuous conduction is happening in unmyelinated fibers. It's really a step-by-step depolarization that's taking place the full length of the axon, whereas saltatory conduction is depolarizing only at the nodes of Ranvier. Let's show you what this looks like. So on the left-hand side here, we have continuous conduction, and on the right-hand side, we have saltatory conduction. We have a neuron, right? We have dendrites, cell body, nucleus, axon, and the axon terminal. This is 1 millisecond, 5 milliseconds, and 10 milliseconds. And you could see in this unmyelinated neuron, at each step of the way, we have sodium rushing in, we have like depolarization happening at each step of the way along the axon. And in around 10 milliseconds, look where the action potential is during continuous conduction. Whereas in saltatory conduction, here we have these Schwann cells. Remember, Schwann cells myelinate an axon, and impulses travel really quick on myelinated axons. So at 1 millisecond, it looks like the action potential is probably the same. At 5 milliseconds, it looks pretty good. But look what happens at around 10 milliseconds. The action potential is already at the end of the axon terminal, where in continuous conduction, it's not. So saltatory, saltare in Latin, means to leap or to jump. So these Schwann cells in between each myelinated portion, it's leaping. It just jumps from node to node. It's jumping over the node of Ranvier. The unmyelinated portions are called the node of Ranvier. These are the myelinated. So saltatory conduction is much faster. Factors that can affect the speed of the impulses, the larger diameter nerves or axons, if they're myelinated, and temperature. So these are all things that can either speed up or slow down. If it's smaller diameter and not myelinated, it's much, much slower, which takes us into fiber type in terms of diameter. So the larger myelinated fibers conduct much faster impulses due to the size and saltatory conduction. So we have different types of nerve fibers. We have A fibers, B fibers, and C fibers. The fastest ones that we have are going to be the A fibers. They are the largest, and they're also myelinated. Think of what happens if you touch something that's sharp or hot. We get this withdrawal reflex. It's super fast. You pull your limb away from the object. So that's super fast, 130 meters per second. That's how fast it can travel. The B fibers, these are also myelinated, but these are to the viscera, to the organs, visceral, sensory, and autonomic, preganglionic. Here I think of like, let's say, a vagal nerve. Okay, when you're scared or you're nervous and you have to run to the bathroom, or your heart rate goes up, or you're exercising and then your breathing increases, these are B fibers. These are autonomic. The slowest are the C fibers. These are unmyelinated sensory and autonomic motor neurons. These are nociceptors. These are pain receptors, but not the acute type of pain. This is like, oh, it's warm outside, or have an itch, or when you exercise and you feel a burn or cramp in your muscles, or dealing with sensual type of touch. Those are C fibers. Can you look at the speed? You could see the A fibers are the fastest and the C fibers are the slowest. In terms of resting membrane potential for muscles and nerves, when we compare them, resting RMP for a nerve is minus 70 millivolts. For muscle, it's around minus 90, and that's for skeletal and cardiac. It's a little bit closer to minus 90. The fastest nerve conduction velocity is 18 times faster than the velocity over skeletal muscle fibers. In terms of synapses, the signal transmission at synapses, we have electrical synapses and chemical. Electrical synapses are dealing with gap junctions that connect cells and allows that transfer of information to synchronize the activity of the group of cells. That's electrical, whereas chemical, it's a one-way transfer of information from a presynaptic neuron to a postsynaptic neuron. So here's a neuron. These are the dendrites. Here's the cell body and the nucleus, and here's the axon. So how does information get to the neuron? Look, we have axodendritic connection. Here's an axon, here's the dendrite, so you have an axodendritic. Here is an axosomatic, because it's going from an axon to the soma, axosomatic. Here's an axoaxonic, going from an axon to the axon hillock. So you have these different type of relays or connections. In terms of chemical synapses, here on the left-hand side, this is presynaptic, and on the right, postsynaptic. So as the action potential reaches the axon terminal at the end, we have the synaptic end bulb. And at the very end, we have these vesicles that can contain neurotransmitters. And maybe it's acetylcholine, let's just say. So calcium comes in, it influxes. When it influxes, it stimulates the release of this acetylcholine, this neurotransmitter, through exocytosis. And now this acetylcholine is released in this synaptic cleft, which is the space between presynaptic and postsynaptic. And the acetylcholine, this neurotransmitter, this chemical, is binding to this channel. When it binds to the channel, it's like a sodium channel. It opens, sodium rushes in, creating an action potential. That's why it's depolarizing on this side. Okay. So again, just another look. Here is the nerve impulse or the action potential coming down to the very end where you have the synaptic terminal or the synaptic end bulb. You could see that we have these synaptic vesicles that contain some sort of neurotransmitter, but calcium influxes in and moves the synaptic vessel to the membrane. It opens up, releases it through exocytosis into the synaptic cleft. Now the acetylcholine is binding to the postsynaptic side. And now this is a ligand-gated channel, because ligand-gated, it's responding to a chemical or a neurotransmitter. And now acetylcholine is the key to the door. It opens up the lock, and now we have sodium that rushes in, and that creates the nerve impulse. The effect of a neurotransmitter can either be excitatory or inhibitory. A depolarizing postsynaptic potential is called an excitatory postsynaptic potential, and that's going to happen through the ligand-gated sodium channels for opening. That's because threshold was met. An inhibitory postsynaptic potential is called an IPSP, and it results from the opening of the potassium channel. And that's going to cause the postsynaptic cell to become more negative or hyperpolarized, right? It's going from -70 maybe to like a -90 on that chart. The further away you move from resting membrane potential in a more negative direction, the less likely you are to reach the threshold, and it becomes more inhibitory, less likely to become excitatory. So excitatory postsynaptic potentials, we're dealing with depolarization. Inhibitory is when we hyperpolarize. Now, a postsynaptic neuron can receive many signals at one time. And neurotransmitters at chemical synapses can either cause an excitatory and inhibitory graded potential. Some neurotransmitters act as the accelerator. Some neurotransmitters act as the brake. So these neurotransmitters could be an on or off switch. And after they do their job, we don't want the neurotransmitter lingering around anymore. So how do we get rid of the neurotransmitter? Well, it can happen through diffusion, where things move from an area of higher concentration to lesser, it just diffuses, or it degrades. So if you have acetylcholine, the body can create acetylcholine esterase, and it just degrades that and breaks it down. Or we can reuptake them. So you could repackage them. So these glial cells can transport them back and reuptake them. Now, we have certain drugs on the market that can block the uptake. So in certain instances, we may want serotonin lingering around here a little bit longer, having an effect to make a person feel happier. So instead of degrading them and breaking them down, people could use, let's say, Prozac, which is a selective serotonin reuptake inhibitor. We call them SSRIs. So Prozac is a serotonin reuptake inhibitor, meaning instead of repackaging it and putting it back into the presynaptic side, it allows it to linger in the synaptic cleft, having a longer effect. Those are SSRIs. In terms of the speed of impulses, we have spatial summation and temporal summation. So let's take a look at what the difference is. Look at the pictures alone, right? So if you look at here and we focus here, we have many presynaptic neurons firing on one, and over that's spatial summation. And in temporal summation, we have one presynaptic firing on the postsynaptic, but this can fire over and over and over again. So the difference here, this would be like all of my students, 50 students saying, I don't want the test today. And they scream it out. Whoa, okay, I guess they really don't want the test today. Here, this is one student over and over. I don't want the test today. 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 So having one fire over and over and over in relationship to many screaming the same message at one time, which is going to stimulate depolarization or hit threshold faster? Well, let's compare. Here, we have some graded potentials, and threshold is met right around there, right? So around, let's say, 35 milliseconds. Over here, one repeating the message over and over, keep firing over and over and over again. By the time threshold was met, it was here, it was around 60. So the spatial summation is the summation of effects of neurotransmitters released from several end bulbs onto one neuron. That's much faster than the summation effect of released from two or more firings of the same end bulb in rapid succession over and over and over. So in terms of temporal summation or spatial, it is going to help you reach threshold much, much faster. So here's the summation effect on the postsynaptic, right? So you have a presynaptic. This looks like it's an excitatory postsynaptic potential. Here's an inhibitory. Here's an excitatory. Here's an inhibitory. Here's an excitatory. So what's going to happen is we've got more excitatories than inhibitories. So the excitatory postsynaptic potential is going to win. All right, it's going to look at the summation effect of many firing at one time compared to one in, repetition over and over and over again. So the neurotransmitters are going to have an impact on excitation or inhibition. And when we come back, we'll get into some of the neurotransmitters.